Thank you very much, Christian, for the introduction and the invitation. It's my first visit to Bonn. I am very pleased to be here. Today my talk will be on the changes of microglia properties after status epilepticus. So I think that I don't have to introduce very much here what is a microglial cell, but briefly just to mention that those cells, which are the resident macrophages of the brain, orchestrate the inflammatory reaction which occurs in many cases after an injury and in many neuropathological disease. The important thing is that the microglial cells can differently influence the outcome of these diseases. And they can have both beneficial or detrimental effects on the survival of the neurons. So this has led to revisiting the idea, the concept that we have of what is the activation of microglial cells. So for many years, we've been thinking of microglial cells having either a resting or dormant states in the healthy brain, and having a ramified morphology, whereas under pathological conditions, those cells will be driven to stereotype, phenotype remodeling towards fully activated states where they have an amoeboid form. So we know now that the picture is a little bit more complicated because even in the healthy brain, those cells are in fact not really dormant, but they are of course very active. And what you see here is a picture from the work of Hemschen and Kirchhoff showing that those cells, in red you have the processes of those cells that have been retracted, and in green those that have been extended during a few tenths of minutes of a movie done in vivo with 2-photon microscopy. So those cells are permanently probing for their environment, looking for warning signals, probably. So, in addition to the fact that they're more in surveying state than in resting states, we know now that when they are activated in pathological condition, depending on the nature, the intensity, and the context of the triggering stimulus, those cells there will be a phenotype selection which will drive those cells in different possible activated states. And then, depending on the duration of the triggering stimuli, then each of these activated states probably will push those cells into different activation states. So, if this hypothesis is true, then we're in trouble, in fact, because then we cannot, or it's really difficult to predict from what we have known, what we have learned from different models of activation, what is actually the activation state of the microglial cells you're interested in a particular disease. In addition to that, we have only a limited number of functional studies of microglial cells in well-defined and physiological and pathological conditions, and most of the work concerning the electrophysiological and the pharmacological properties of those cells have been done in culture system where those cells are already half activated. So the good news is that we now have some transgenic mice that we can use to visualize and identify those cells in vivo, in the whole brain, in the living animals, or in acute slices prepared from these animals. And the one we have been using in my lab is the CX3CR1 EGFP heterozygous mice developed by the lab of Littman. And this has allowed us to target microglial cells in acute slices of the hippocampus and to look at their electrophysiological properties, their pharmacological properties, and their motility in the slices. So we've been interested in epilepsy basically, originally because of the fact that in many models of epilepsy and as well as in human epilepsy, there is a inflammatory reaction which occurs. Here it's an example of experimental temporal lobe epilepsy in mouse with an injection of kinates. And this inflammatory reaction is quantified here according to the level of expression of several inflammatory molecules that very rapidly after the triggering of the initial status epilepticus by the injection of kinates increase a lot their expression within a couple of hours, in fact. Some of them have their expression returning to baseline, some other remain at a high level of expression for many days. So this inflammatory reaction is accompanied by, both in human and in experimental temporal lobe epilepsy, by the fact that we can observe reactive astrocytes and reactive microglial cells in the hippocampus. So what we wanted to do is to look at the microglial cells here, because, of course, one hypothesis is that these reactive glial cells might become reactive because there's a lot of neuronal death. And so it's the death of the neuron that could trigger the activation of the microglial cells and of the astrocyte later on. But there is another hypothesis that would be that, in fact, the hyperexcitability of the neurons that occurs during the status epilepticus can instruct the microglial cells that something wrong is going on and that this hyperexcitability will turn microglial cells to an activated state that, in turn, could lead to the remodeling of the network and the neuronal death that eventually will lead to the occurrence of true recurrent epilepsy. So our working hypothesis is that it's the activation of, or at least that the activation of microglial cells by releasing different types of mediators, by phagocytosis, by going around the neurons, can in fact change the network properties. So we have not yet identified those mediators. What we have done instead, as a first step toward this study, is to look at the functional properties of the microglial cells, aiming that identified some key molecular elements that could be important to trigger this phase of during the latent phase of epilepsy. So we have been using a very simple model of status epilepticus, namely intraperitoneal injection of kinates that leads to a status epilepticus, and then at different time points, then we sacrifice the animal and either look at the expression of different markers in the brain or prepare acute brain slices, acute hippocampal slice, in which we can identify the cells thanks to the fluorescence of the microglial cells expressing the GFP under the control of the fractalkine receptors, and do some phenotyping of the microglial cells. So we first checked that in those mice, we indeed have an inflammatory reaction, and these are the results of real-time PCR from the whole hippocampus, where you can see that as early as 3 hours after having induced the status epilepticus, you have a 10-fold increase in the expression of EL1 beta, EL6, TNF-alpha, and COX-2, so 4 different inflammatory markers, and that this increase is maintained over at least 2 days. We have also characterized the neuronal side of the model, and I just show you here some fluoro-J-B labeling, which is an indicator of cell death, suffering of neurons. And you see that 48 hours after having induced the status epilepticus, we have some labeling here indicating that both in CA1 and in CA3, actually, in the hippocampus, we have some neuronal damage. So now we look at the microglial cells in these animals. This is the normal morphology of the microglial cells in the CA1 region of the hippocampus, in the striatum radiatum. So these are control animals, GFP, immunolabeling with IBA1, which is a specific marker of the microglial cells, perfect colocalization of the two, of course. And then 24 hours after the status epilepticus, you see that the morphology of those cells are completely changed. We have an enlargement of their soma, a thickening of their processes, and a retraction of the thinner processes. So this was actually known. We were expecting something like that. We quantify the numbers of microglial cells, and you see that their number increases after the induction of the status epilepticus. And by 2 days after the status, we have roughly a doubling of the number of microglial cells in the CA1 region of the hippocampus. So this could be due to migration and proliferation. So we have at least checked that those cells are indeed, some of them are proliferating. So this is a double labeling of GFP together with Ki67. And you see that after 24 hours, we have some labeling, more evident here at 48 hours, that colocalize of this proliferative marker with the GFP of the microglial cells. We also use the MAC2 antibody, and MAC2 is expressed in activated microglial cells that have entered the proliferative state. So, and here again, we see we see the colocalization of GFP and MAC2. So we have some proliferation in the hippocampus, more microglial cells, they are bigger. We do not exclude that there could be some recruitment of microglial cells in addition to the proliferation that might explain the doubling of the numbers. Okay, so far so good. We have a model in which we do reproduce the activation, let's say the morphological activation of microglial cells, so then we can start to look at their functional properties. So the first thing we looked at are some basic membrane properties of those cells, passive properties. The capacitance, the membrane capacitance, which is an index of the total surface of the membrane of the cells that you record with patch clamp, in our case with patch clamp recordings. And you see that when compared to the control animal injected with PBS, though they have received a KNH injection, after 1 day, they have an increase in the capacitance, in the membrane capacitance, which is in good agreement with the fact that those cells are becoming larger. Okay? At the same time, we have, in fact, a drop in the input resistance, which is an index of how many open channel those cells have in their membrane. And in fact, this indicates that they have more and more channels in the ion channels in their membrane, and mostly potassium channels that will drive the resting membrane potential of those cells toward more hyperpolarized values. So if we look now at some more voltage-activated properties, this is how it looks like the IV curve of microglial cells in the resting hippocampus. Rather boring recordings for an electrophysiologist. There is almost nothing to see. It's only passive membrane properties. Whereas 2 days after having induced the status epilepticus, we start to see potassium currents that are activated by hyperpolarization and some others that are activated by depolarization. So if we do those experiments at different time points, you see that 3 hours after status epilepticus, we don't have a big change. The green, the black, sorry, is the control. The green is the animal 3 hours after the status epilepticus. The blue is 24 hours. And you see that at 24 hours, we have an increase of those inward currents and a small increase of the outward currents here. 48 hours after the status, we have a decrease of the inward currents and an increase of the outward currents. And in fact, this sequence of high inward currents and then a decrease of them recapitulates what has been seen in culture when you put those cells, I told you that when you put those cells in culture, they have already half activated, and the property that they have in culture is the blue line, basically, the blue IV curve. And then if you treat those cells with LPS to fully activate them, then you end up with the red one. So by looking at different time points, in the slices, I mean in the brain of those animals, we actually see this sequence of events, the beginning of the activation and then the full activation of the microglial cells. So we characterize a little bit these currents, these outward currents, because there have been some data in the literature showing that, in fact, different potassium subunits could underlie these currents, and probably those subunits might be associated with different functional properties of the microglial cells. So the most likely subunits that can be found in microglial cells are those ones, Kv 1.1, 0.2, 0.3, 0.5, or 3.1. So we did some basic electrophysiological characterization of those currents, their activation and inactivation curve. We also looked at the way those currents inactivate upon repetitive activation, and you see that when you start to stimulate those cells at higher frequency with depolarizing pulse, you have an inactivation of the current. So you see here also that at steady state depolarization, the outward current inactivates. Then we did some pharmacology. The current is fully abolished with 4-AP. It's more or less, it's a little bit inhibited, but not that much with high concentration of TEA. It's not affected by, or just a little bit by dendrotoxin, but it can be inhibited by high doses of agitoxin and low doses of margatoxin. And this is typical of the pharmacology of Kv 1.3 subunits, which has been linked to, in fact, the proliferation of microglial cells, as well as the fact that those cells can actually, through the NADPH respiratory burst, release reactive oxygen species that might be deleterious for neurons. So this is the subunit that we have here, and which might be linked to the proliferation of microglial cells that we see. So then we turn toward the purinergic receptors that those cells might express. Why? Well, it's known that microglial cells can express P2X4, P2X7 receptors. Those receptors are ionotropic receptors, so channel receptors which are in fact ion channels. P2X4 has been shown to be important for the release and the secretion of BDNF in the spinal cord from microglial cells, whereas P2X7 is important for the secretion of interleukin, and in particular interleukin-1 beta. These two receptors, P2Y6 and P2Y12, are in fact metabotropic purinergic receptors. The first one is important for the phagocytosis function of microglial cells, whereas this one, P2Y12, is important for the process extension and the polarization of microglial cells that will occur when those cells are activated, and I will show you some examples later on. Interestingly, during activation, when microglial cells are activated, the first three receptors are upregulated, whereas P2Y12 is supposed to be downregulated. So we looked at the expression of those receptors by qPCR in the whole hippocampus, and we were surprised to see that, in fact, all messengers for all of these receptors were upregulated. So it's true for P2X4, P2X7, P2Y6, and P2Y12, which was a kind of a surprise to us. So then we decided to record directly from microglial cells and to test for the functional expression of these purinergic receptors. So this is a recording from an hippocampal microglial cells in a healthy brain, in a control animal. And you see that upon application of a saturating dose of ATP, you have a tiny current in those cells, few picoamps, that you can record because the input resistance of those cells is so high that you can detect very small currents. If you do the same experiments in microglial cells in a slice of an epileptic animal, this is the type of response that you have. And on average, these responses are bigger than those ones. So these are the group data, where you see the IV curve of the response of the group data for the response activated by these inotropic receptor mediated responses in control animal and in epileptic animals. And you see that we have an increased conductance in these responses. So these responses are mostly due to the activation of inotropic receptors, because we managed to block those that can be mediated by metabotropic receptors. So then we try to identify which of the receptors could be underlying this response, and we use a trick that has been used to potentiate the responses due to the activation of P2X7, which is to remove calcium and magnesium from the external medium. This is known to potentiate the response due to the activation of P2X7. And indeed, when we do this manipulation, we have an increase in the density, in the current density that we can record from those cells in controls animal. Look that the scale bar here is 10 times bigger than here. But still, we keep this, the difference between the control and the epileptic animals, mainly that in the epileptic animals, we also have a potentiation of the response when we remove calcium and magnesium, but the response is still bigger than here. So basically what we have here is a response which is mostly mediated by P2X7 receptors and which is potentiated in the epileptic hippocampus. So this is the same data than here, plotted in another way. So this is the average current that we can have in control animal, the doubling of the response in the epileptic animals. These responses are both in the control and the epileptic decreased by this compound BBG, which is an antagonist of the P2X receptors and mostly of P2X7 receptors, although it's not so clean. We did some experiments using a P2X4 knockout animal, and we didn't see a significant difference between this knockout animal and the wild type animal. So then as I told you, we can increase the response in the wild-type animal by removing calcium and magnesium. This is the data. And this response can be blocked by the highly selective P2X7 receptors, these compounds synthesized by Abbott, that wiped out the response both in the control and the epileptic animal. We had access to a P2X7 knockout animal, and basically we reproduced the effect of using the antagonist. Basically, we wiped out the P2X receptor-mediated response in the microglial cells of both control and animals. So the conclusion of these studies is that in our conditions, the main inotropic receptors, purinergic receptors of those cells is P2X7. So then we looked at the metabotropic purinergic receptors. And so we set up the recording conditions so that we would not have to bother about the inotropic receptors too much, but focus on the potassium currents that can be activated through protein G-coupled receptors, purinergic receptors, to the response of those purinergic receptors. So we use UDP as a selective agonist of P2Y6 receptors, and you see that we can see an increase in the potassium conductance. You have the IV curve of the response here. And when we do the same manipulation in epileptic animals, then we have an increase of this P2Y6 receptor-mediated response, and these are the group data. So P2Y6 is upregulated in the epileptic animal, and we did similar experiments with this compound, which is selective for P2Y12 and P2Y13 receptors in microglial cells, and you see that we also have an upregulation of the P2Y12 or 13 receptor-mediated responses in this cell. So we were, in fact, a little bit embarrassed because, as I told you in the beginning, this receptor, P2Y12, was supposed to be downregulated in models of microglia activation. So this is a paper published by the group of David Julius some 3 years ago now, where it could show that either when injecting LPS, which is a strong microglia activator, in the brain, or by using a model of traumatic brain injury, you have, in fact, an activation of microglial cells. So these are the LPS model. and that the immunostaining for P2Y12 is completely abolished in these states of fully activated microglial cells. This is the same result, but looking at the messenger's RNA instead of looking at the protein. So we obtained the antibody from David Julius, and we did the same experiments on our preparation, on our model. And you see that in the control animal, we have a nice labeling of P2Y12 restricted to the microglial cells. But then upon activation of those cells in the epileptic hippocampus, we still see a strong immunostaining in activated microglial cells. So because this receptor is supposed to be important for the motility of the process of microglial cells, we in fact study the progression of these processes towards a source of ATP or an analog of P2Y12 receptors. So we put a patch pipette containing the analog for the agonist for the P2Y12 receptors. And what you see here is that the processes of microglial cells are going toward the tip of the pipette, which is located here under the red dot here. And within three quarters of an hour, they really reach the pipette. Okay? So now if we do the same type of experiments in a slice from an epileptic animal, you see immediately that things are going faster. Okay? So first you notice that you have more microglial cells, and the migration of this wave of fluorescence toward the tip of a pipette is going faster. So we quantified this effect by comparing the change in fluorescence in the whole field of view and against the center. And this is this plot here. So the control, you have an increase in of fluorescence in the center of the field where the pipette is releasing this analog of ATP. And you see that the increase in fluorescence in the center is going faster in the epileptic animal. So then we measured the speed at which this wave of fluorescence travels toward the center of the field. And this is done here by measuring the fluorescence at different concentric rings in the field of view. And you see here how the waves goes through rings that are more and more close to the center of the field. And by knowing the distance between each ring and the distance between each of these peaks, you can derive the velocity of this movement, of these waves of fluorescence. And this is what is plotted here. We have a doubling of the velocity of the processes of the microglia toward a source of ATP. So this confirmed that not only we can detect an upregulation of the messengers of P2Y12 receptors, but we have also an increase of the response induced by the activation of those cells, either when we measure the potassium currents that are linked to these receptors or when we measure this functional the motility of the processes. So in contrast to other models, some other models of activation, we see, of course, an upregulation of P2X7 and P2Y6, but we see also an increase of P2Y12 instead of seeing a decrease of these receptors. So it's the first, actually, evidence then the activation state that we have of these microglial cells in the epileptic brain is different from that we can have in for instance, the very widely used model of LPS. So what about P2X4? In fact, when we started our project, it was to study this receptor, which seems to be not so important in our model of epilepsy. I have to tell you that in models of neuropathic pains, the release of BDNF, which requires the expression and the upregulation of P2X4 in microglial cells, is responsible for the allodynia that can be triggered by a lesion in peripheral nerve. So if you block P2X4 P2X4, if you work with the P2X4 knockout animal, and if you do a lesion in the peripheral nerve, the animals will not have any increase in the pain that they feel compared to the wild-type animal. And so we were expecting to have an upregulation and a strong effect of the knockout, of the invalidation of this receptor in the model of epilepsy. But instead, we have, in fact, no real phenotype of P2X4 knockouts in the model of epilepsy. So this is the similar type of experiments that I just showed you before, because P2X4 was also proposed by some authors to be important for the polarization and process extension of microglial cells. In fact, when we compare the knockouts to the wild type, either in the control condition or in the epileptic brain, we see no difference between the two phenotypes, the two genotypes. Sorry. But we have some slight effect of P2X4 receptors invalidation, and this is the membrane capacitance that I told you increase quite a lot in the wild type animal. It's still increasing in the knockout, but a little bit less than in the wild type. We also have a deficit in the expression of these potassium channels that has been proposed to be important for migration of microglial cells. So we might expect a change in the number of microglial cells that are present in the hippocampus. And some preliminary data seems to indicate that this is indeed the case. So to finish, I would like to present you these preliminary data that we have obtained by comparing the P2X4 and the P2X7 knockout mice. And as you can see here, these are some qPCR data showing that some of the expression of the inflammatory markers that I presented at the beginning of my talk. So IL-1 beta is, as I told you, increasing the red, if you follow the red bars in the wild type. There is not a significant effect of this increased expression in the knockout for P2X4, but if you look at the P2X7 knockout mice, we completely wiped out the increased expression of this inflammatory marker in the hippocampus. And the same holds true for interleukin 6. BDNF is a little bit affected by both actually knockouts, but not that much, not as strongly as the two interleukins. And TNF-alpha is also a little bit affected. In fact, it's still increasing, and there is no significant difference at 2 days after status epilepticus, but it seems to be a little bit delayed in the increased expression. So, in contrast to what has been shown in the spinal cord using model of neuropathic pain, the invalidation of P2X4 doesn't have a major effect in the inflammatory reaction that we can see in the hippocampus of the epileptic animals, whereas invalidation of P2X7 receptors has a dramatic effect in the development of the inflammatory reaction. So this is where we are now, and by looking at the neuronal changes that occur in this animal model, we expect to be able to identify some important markers, and of course we will focus on the signaling of IL-1 beta to see how microglial cells can in fact impact the activity and the survival of neurons. So, in conclusion, our data show that after status epilepticus, you have a very strong activation of microglial cells, which can be seen as well as by looking at the morphological changes of those cells, but also by looking at the functional properties of those cells, we have a strong activation, a strong upregulation of voltage-activated potassium channels. We have a strong upregulation of the receptors that mediate all the responses due to ATP or UDP in those cells. And contrary to other models, we have some different, I mean, we have only a minor phenotype of invalidating P2X4, and we don't have a downregulation of P2Y12. So our data are fully agree with the hypothesis that different stimuli will induce the development of different activation states of microglial cells in different models of activation. And this is what remains to be done, is to understand how these specific activation states can impact the neuronal survival and the neuronal rewiring that occurs in epilepsy, especially during the latent phase of epileptogenesis. So this is the people that have done the work that I have presented to you today. Elena Avignon, who really developed all the model of epilepsy and the study of microglial cells in my lab, helped with Françoise Levavasseur, who did the qPCR analysis, Alexis Maintain, who did the study of the potassium currents. This work has been done in collaboration with François Rassendren in Montpellier and Lauriane Ullmann, who produced the P2X4 receptors and did some of the immunostaining. And these are Cathy Vantalon and Valentina Emiliani, are two physicists working in the lab next to ours to develop microscopy, new microscopies. And I just show you one of those movies where we could have some kind of cheap but quite nice quasi-confocal pictures of microglial cells by using some different optical tools. Thank you very much for attention.